but I wanted to welcome you to our April 17th webinar that's sponsored by the Water Modeling Work Group at EPA. Just so that you guys know, the EPA Water Modeling Work Group, these series of webinars are meant to help water quality professionals better understand surface water quality modeling and how models can be used to solve common problems that face water quality regulators. And I do want to offer the introduction to presenter to my colleague in the USGS, Leave.
Yes, hi, Anthony. So Anthony and I work as part of the Lake Champlain Federal Partners Working Group, and we have been talking about Lake Champlain, and I have a hydrodynamic model of Lake Champlain, and we're have been interested in extending it into water quality modeling, and so we thought that this would be a great opportunity to introduce or to explore some of those possibilities with the Deltares software, which is how I built the hydrodynamic model. So we've invited Dr. Markus here. He studied physics at the University of Delft and then started working at Delft Hydraulics quite some time ago. And it's now Delft Hydraulics is Deltares. He's been involved in water quality modeling for almost 40 years. He did his PhD at the University of Amsterdam almost 10 years ago, studying the transport of fate and nanoparticles. And I think I first met him when he was offering a course on this water quality modeling software, and then I've had the pleasure of working with him on implementing some residence time calculations in San Francisco Bay Delta in this modeling software, DelWalk, that you are going to hear about in the seminar. So take it away, Arjen.
Okay, thank you. Well, let me first share my screen. What I'm going to talk about is what we call the water quality. It's part of a larger software system, and we use it for all kinds of water quality modeling. Quite often in combination with the hydrodynamic model, which is part of this system, but it can also be used in other contexts with other hydrodynamic and hydrological models. The name will vary over this presentation because I happen to call it DelWalk. It's the old name. It's the name of the program which is beneath all this, and the water quality is this computational program together with the user interface and everything that comes with it. But let me start with the just a general introduction of the modeling suite. Let me get a nice pointer. Right. Okay. This modeling suite uses unstructured grids. Here's an example. A small part of a model will be showing more about later on, and show that it's really an unstructured grid. Here is a grid based on mostly triangles, but also some more complicated forms. And in this case, I took the liberty of using an aggregated version of that grid, so that you can see that the water quality, though it always takes up the grid from the hydrodynamics, it can also do some variations which are often useful because water quality in general requires less resolution than hydrodynamics does. So often we want to make a coarser grid based on the hydrodynamics and use that for our calculations. But to get back to Delft3D FM, so the whole modeling suite. The things that are included are hydrodynamics. The hydrodynamics of coasts, lakes, rivers, that sort of areas. Morphology. So the development of the riverbed, for instance, or in tidal situations, also the formation of gullies and that sort of bed forms. Surface waves, because they are part of both the hydrodynamics and of morphology. Water quality in the classical form I'm going to present here, using the advection diffusion equation and particle tracking as a different way of solving water quality problems. Like I said, the hydrodynamic model is used to drive the water quality model, and in general, we first have the hydrodynamics and then use that following the water quality model. So that's basically what we call the offline coupling. It allows us to couple not only to Delft3D FM, but also to all kinds of other hydrodynamic and hydrological models. In this case, I'll be presenting results using Delft3D FM. So to go into the water quality part a bit, what the water quality provides is, well, first of all, the offline calculations. So you have results from the hydrodynamics, and you use that to drive the water quality calculations, and you can reuse that hydrodynamics all over. Because that's done, it's stored, you can use it time and again for all kinds of calculations. Something that's been becoming more popular the last few years is that we also can include the water quality processes in Delft3D FM itself. That means that during the calculations of Delft3D FM, you also have the water quality calculations. They go along, and it saves you storing the dynamics, which can be very large. For instance, we have fine resolution models of the North Sea. It could also be one of the Great Lakes in your continent. And you can imagine that if you have a fine grid and you calculate for a long period of time, the files that result may be very large, and they become at some point unmanageable. So that's where online calculations come in. The drawback of that is that you have to do both hydrodynamics and water quality in one calculation. So you always have the overhead of the one or the other. But that's something that you can decide upon per project. In most cases, I work using the offline calculations, and I'm most familiar with that mode of working. In both cases, the selection of the substances and the associated processes is done via the input. So rather than configuring the program itself, we use an external file which contains the definitions of our substances and how they are acted upon by the processes, and we select the substances of interest along with the processes of interest, just as we go for our needs. An example I show here. This is the mass balance for a particular location, and it includes a number of processes like the uptake of nitrate by diatom, uptake of nitrate in a green algae, And CM is it's one of our new models for algae. Those are heterotrophic algae. Don't pin me on this. I'm not an ecologist. But the idea is that these algae, or almost zooplankton, can act as a phytoplankton, but also as a animal, in fact. This is just an example of how these processes work out. And as you can see, this red part, the uptake by CM of nitrate, constitutes quite a large contribution. And the transport into this column, this location, that is compensating that uptake. This is one way of looking at mass balances, and it teaches you how important processes are, and you can see whether processes need more attention, or if they are so small that you might almost leave them out. Those are all things you can derive from such mass balances. They are produced if you want. If you don't want them, they won't be showing up. But what I'm particularly proud of is that any process parameter that appears in our processes can be a constant, a time series, or something that varies over the model area without you having to do much more than defining it as such. So the program takes care of all the details, and quite often we want a parameter to be a time series or even vary over the whole model area. And in other cases, it's simply a constant, and you don't have to do much about that. Output, very typical, map of the whole region, detailed history for selected locations of areas, and these mass balances if you ask for them. Another thing which is quite of interest is that we can also define statistical output on this, on the concentrations, but also on the process results. That's all taken care of via actually the same mechanism as selecting the substances and processes. If you are interested, I can tell more about it, but for now, just know that it's possible. Another nice feature, I think, of the water quality is that besides the connection to different hydrodynamic models, it also can deal with 1D, 2D, and 3D models without having to use a different program. I'm going to show you the user interface we generally use for this. There are other ways to set up the calculations, but certainly for setting up the model, I like to use the user interface. And one of the nice features of that is that you can use fractions. I'll explain that. What we mean by fractions in this case is that all the sources into the system, and if I go back to this picture, this is an open boundary to the sea. We have a small source over here, one over here, I think, and one over here, and here the river is coming in, and all these sources are automatically translated into a tracer that comes in for the river via this river boundary. For Flushings, that's this city, it comes in at this source and so on. The last tracer which is then produced is the tracer to see where the initial water is going to. That's explained a bit here. So all the sources become separate tracers. So that means that you can see what the effect is of a particular source. The ones along the estuary are very small, so quite probably they don't have much influence. The sea boundary and the river boundary, on the other hand, do have a large influence, and you can see using this feature very fast what kind of dynamics there is in the system. And that's what we use it for, to get a feeling for what the system is all about. Perhaps this is a good moment to demonstrate that. Let me just do that. I'll start a new project. Are we over here? And I think I have right. Okay. So now it's loading in the information about the grid, and to show you where that is actually located. I think if I move this around, yep. I am located over here. So this is an estuary somewhere in the south of the Netherlands, on the border with Belgium. And I use this as a demo model because it runs quite fast. Within a few seconds you have a result. To set up a calculation with these fractions, I generate them. And if you look at the initial conditions, you'll see that there are indeed a number of traces here. The sea boundary, the river boundary, 3 small sources, and also the initial water which is present. And that has a concentration of 1. I can now generate the boundary conditions for this. That has been done, and I just run the model. And I show you what results here. Let's take the river boundary. We put up the map can. So you see the water from the river, from the open sea, entering the estuary, slushing back and forth due to the tide. And you can also see that, oops, where is my cursor? Over here. You can also see that it will come up to about this area, and the other half of the estuary that's dominated by the river. So in this way you can get a quick feeling for what kind of dynamics we're talking about in this particular system. And this is just to show that how this feature works. And of course, if you load in a different substances file, like over here, these are a few of the predefined ones, but you can also load in one of your own. And then you get a different set of substances with the processes that belong to that, and you can set up a completely different calculation. Well, that concludes the demonstration of these fractions. Let's move on to, well, the typical kind of applications we have here. Algal growth and eutrophication are things we quite often deal with. In North and in a sea area like the North Sea, we have quite some input from rivers. So even though the eutrophication itself has gone down over the decades, we still have some problems there. Sometimes local, sometimes much more complicated. We also use this to study the effect of wind parks in the North Sea. We have an operational system in Singapore, for instance, where this kind of things are done. To give some details, algae can be modeled in 3 different ways. We have a simple model which distinguishes green algae and diatoms. It's fairly simple to use and simple to understand, but it's limited in its capabilities. Then we have the Bloom model, which is capable of dealing with multiple species and multiple groups of species. I have a webinar that you can look at later on, which explains the principles there. And the new algae model is called Protist. It is about green algae and diatoms, but also the heterotrophic algae that exist, and zooplankton and things. It requires a bit more input, and that may or may not be difficult to handle. We think it is a nice one to if you have a need to understand what can be happening with different trophic levels. So that's the latest development in that respect. Another typical application involves sediment transport, and there we have to deal quite often with dredging and dumping. So material is dredged at one point and moved to another point in the whole system. But sediment transport, well, a plume like this is also dealt with within the water quality. Typically, toxic substances are heavy metals, pharmaceuticals. We deal with them more and more. PFAS has been a topic for the last, well, at least 10 years now. And this week we heard that eggs of chickens that people keep in their gardens contain so much PFAS that it's not good to eat these eggs. So within the Netherlands, PFAS really is a problem. Bacterial pollution, I understand in Lake Champlain, that's certainly something to be looked at. Again, for the water quality, that's one of the sets of substances we distinguish. It's also used for complete environmental impact assessments. So in that case, you have a large number of scenarios to look into, different kinds of substances in general, and look at the consequences of these scenarios or these waste loads, etc. The water quality is then one of the vehicles to do the analyses, and of course you have to draw your own conclusions from these calculations. There are many more possibilities, and it depends a bit on what you want to do. But know that there's a lot of stuff already included in the library. Here a bit about the Bloom module. This picture shows you the development of chlorophyll over time, and what I hope you can read it. This part of the graph shows you what the limiting factors are for the growth. And for instance, this yellow line shows that phosphorus is limiting for a large part of the year. Mortality simply means that they can't grow fast enough, or at least they cannot grow more abundantly. Below here is a typical succession of the alcohol groups. I don't know too much about these groups, so I can't really explain them. But you can see flagellates, phaeocystis, various groups of diatoms, etc. So Bloom is indeed capable of predicting this sort of successions of algae. And here is a link to the webinar that you can watch if you are interested in this. It's a bit long, so I'm not going to show it you right now. I can put in the chat the link, so you can have a look later on. Next one. Right. Well, I've already showed you the very quick demo about the fractions. What I want to do now is a slightly more involved demo. It's actually a calamitous spill that happened a couple of years ago in the River Scheldt. And if you go back to this project again, the River Scheldt is running over here. It has a number of tributaries, and here, after passing the city of Antwerp, it enters the Western Scheldt, so this estuary. The calamitous spill occurred in the Scheldt River itself, and it consisted of a large amount of sugar beet pulp entering into the river during a very short period of time. So a large amount of organic material causing depletion of oxygen, and literally thousands of fish died, and there was oxygen depletion over a large area for a fairly long time. So for this demo, I could have put it into the river itself, but it makes a better picture if you put it into an estuary. So that's what I did. I estimated the amount of organic material because it's organic material acting on oxygen. I chose a set of substances which is limited to that. You need to select, of course, a location where this occurs and some period of time. It's not meant to be an accurate mimic of this particular accident. It's just an illustration of how you can model this sort of things. Before I start, just a bit of theory. And that theory is actually very indicative for all these processes, well, for many of the processes we have within the water quality. So we select BOD and oxygen as the substances, and we distinguish actually two processes only. Decay of BOD, which consumes oxygen, and oxygen can be entering the water again from the atmosphere via reaeration. This picture illustrates what that would typically look like if you did nothing. Actually, it's a part of an experiment I did a couple of years ago regarding joint modeling. That's a very different technique than what I'm going to show you now, but I had the picture ready, so I thought to reuse that. If you want to know more about that, we can talk about it. Okay, so typically what you see is that BOD will decay over time. At first, oxygen will be depleted, and then due to reaeration, it will restore itself again to the original values. That's what's happening in a well, if you take a beaker of water with a lot of BOD in it, it will also happen on a larger scale in an estuary like the one we're going to get. So what I'm going to do is show you how this works. Let's see. I'll get a new set up a new project, and I need to have back. Yeah. So, as you can see, or at least let me tell you that it's so. This is a finer model which extends much further into Belgium, and it also extends much further into the sea, into the North Sea, in fact. We've been using this as a part of the transport timescale project Liva already mentioned. It's larger, so a calculation will take longer. But what I'm going to do is just show you what the steps are, and then I'll bring up a calculation I did before to show you what results. So I want my own where is it again? So I now have these substances, oxygen and BOD. I'm going to take the initial value of oxygen to be 8 and BOD, since it's only entering via the via this clamp spill, I leave it to be 0 all over the place. Oxygen will be 8, because that's more or less the saturation concentration. Boundaries, well, there's quite a lot, and I need to have boundary conditions for this. So I prepared that, and I'm going to import that. That's this stuff. Yep. Okay, and I also need to add a load. So let me see what's here. I'll add a load somewhere over here. For this demonstration, it doesn't really matter where, but this is one way of doing that. And I'm going to call it spill because that's the name I've used in the file I've prepared for this. Loads data. So what I have is that the BUD comes in at a very high amount. It's a block function of time, so it starts off at 800,000 grams per second. It lasts 1 day and then it stops again. There are various ways of defining these waste loads within the water quality, but this is in the principle, and now I could start the calculation. It's just a calculation which lasts some 20 minutes. So I'll open the result from the previous calculation. Let's see, that's this one. And when I show oxygen, what is a bit awkward about this one is that the color scale is the default color scale is wrong for oxygen. I would like it to be blue rather than red for the high concentrations. So what you see is that. Let me bring back the background again. I'll zoom in a bit. So what you see is that in the neighborhood of where we had the waste load over here, you get very low concentrations of oxygen. Legend over here. Actually negative because it's a naive calculation. But it indicates that there's a lot of organic material that consumes oxygen, and you see that it will extend over a very large area. If we instead look at BOD, again, let me get the map to, and then over here. It's spreading out over a large area. And of course, not always the oxygen concentration will be very small or even negative, but it is certainly an amount of organic material which influences a large part of this Western Shelf. Well, the data I used were very rough. I had to estimate the amount of organic material these sugar beets represented. There were no hard figures in the articles we found about it, but it does illustrate how you can set up a calculation to analyze this sort of things. Okay, well, that concludes this part of my presentation. Let's move on to the next one. I told you about the substances that are already there and all the processes that are involved. We have made no effort to put in everything. And that's not really necessary either, because it is possible to add new substances and new processes without too much programming. Certainly, if the processes in a mathematical way are simple, it's fairly easy to do so. And sometimes no programming is involved at all. It's just a matter of extending the configurations of the processes. For instance, we have a small set of generic decayable tracers, numbers 1 to 5. If you need more, you could simply copy the configuration of one of the processes there, and adapt it for decayable tracers 6 to 20. In fact, I started doing that this afternoon for a client who asked about that. In that case, you don't have to do any programming at all. Just run a few tools and do some editing in this this large configuration file. But otherwise, you can add process routines via what we call the Open Processes library. There's documentation about that. During the courses I gave up to a few years ago, I sometimes was able to elaborate on that. It depended a bit on the time we had. But notice there, sometimes you have to do quite some programming, because the processes involved are complicated. In other times, you may be able to use what's already there. So basically, the message is that you don't need us to extend the program. You can do it yourself via this particular feature. Yeah, well, that concludes my my presentation so far. I would like to open the floor for discussion.
Thank you, Dr. Markus. Well, let's start out with a couple of questions on the mechanics of the model itself. PC based, so it looks like it was Windows, right? What are you using for the GIS? I'm sorry, go ahead.
The user interface I've shown you is indeed Windows based, but the program underneath can be used on Linux as well. And in fact, our larger calculations are done on our Linux Linux cluster, in-house, or even at a large supercomputer center in Amsterdam. So that's not the limit.
Gotcha. And the mapping architecture you're using, is it something you're getting from another program, or is it built into the program you have?
You mean the background I showed?
Yeah.
Okay, that's something that's being done within the user interface. What it does is it uses OpenStreetMaps to show the background. I have no idea how that works.
Okay.
It seems possible to use other sources of maps as well, but I would have to ask my colleagues about that.
Okay. Yeah, it looks very familiar to QGIS, which is why I was asking that question.
Basically, it's a how do you call that? A GPS, geographical presentation system.
Ah,
So
okay.
it basically is the maps that are beneath. So not so much analyses that you can do. What I do understand is that the output from the water quality, but also the flow can in fact be loaded into systems like QGIS because we use the UGrid convention to store our data.
Hmm.
So that's NetCDF with a particular convention to make sure that programs don't need to know exactly what's in there. They can read it by following this UGrid convention. Sorry.
Now, I understand this is an open source model, right? So
Yes.
are you using the GNU license for this?
Oh,
Oh,
he's joking.
oh no.
um, I can answer the listening question, but I can tell you that at USGS we run the software, we've compiled the code in our HPC environment and run the hydrodynamics as well as the water quality program on our USGS high performance computer system called the Nolly. So it's doable, and that now you're just downloading the source code. We do also sometimes use this GUI in Windows as well, which requires a different license. So I think to get anyway, so I can let Arjen answer that question, but yeah.
I think you simply ask for a license. I'm not quite sure what the financial what financial aspects are. I think the license is mainly meant to monitor who's using it. And for me, and for a lot of others, the user interface is mostly suited to set up the model. And then once you have done that and you know where the waste loads are located, etc., you can use the input files which are beneath this to do the calculations that you want. Because it's not always easy to put in a large set of time series, for instance, and then people use scripting tools to prepare these files. Also, I haven't really shown that, but if you use the user interface, then it will run the program underneath and will only stop when the program stops. That, of course, is useful for short demonstrations, but if you have a calculation which lasts a few hours, then your PC or your workstation would be bound up by that. And in that case, I would resort to using the computation program directly, which is also possible. So then it can run in the background and I can do other stuff.
And then there's also requests for the links, webinar links for the modules. Do you you're going to share the links are active in your presentation, correct? And you're going to share your presentation.
Yes.
Yeah,
Let me put in.
I posted the Vimeo link for you.
Oh, right. Okay. Yep.
Okay.
Arjen. So yeah, I posted that. It looks like the right one. You can check. It's called the Delft3D Phytoplankton Modeling Concepts of Bloom. So it sounded like the right one.
Okay. Are there any more questions at this point?
Yeah, no, I can just add too, there's a lot of like through open earth tools and other things, there's a lot of open source tools available to like script the inputs and the outputs and to process the outputs to make maps and things and other
Yes,
on other
there's quite a lot.
platforms.
There's
So,
quite a lot.
yeah.
I normally use our own program, QuickPlot, because, well, being more in the development side of things, developing and maintaining the program, my needs for presenting or making nice presentations is much less than for the people who do the actual projects. But there's quite a lot of stuff available. Open Earth Tools is one thing. People have also been looking at well, Python, other Python stuff. I may be able to bring up a very specific one. Let me see. Let me see. It's not interesting. Just go. Right. This is what I meant. This is actually a result from a monopile, water inside a monopile. We've used Deltares to model that, together with the hydrodynamics being estimated. And what you see is the concentration over the length of that monopile in time. So this sort of presentations is also done. And that's something completely different from the estuary I showed before. In this case, we're talking about a 1D model, in fact, which is driven by the flow coming in through the holes in the monopile, due to waves and due to tide, etc.
Yeah, and I will just highlight that, you know, this model works really well, but it is reliant on the hydrodynamic input, right? So you need to make sure that that is all accurate. To the water quality won't be right if the hydrodynamics aren't right. So you need to, you know, there's
Right, yes.
yeah, this is all built on that, but maybe
I can also I can also show you something completely different.
There's also a question if you can show an example of PFAS work, Arjen.
Right, yes. Actually, that's what I was looking for.
Oh.
Let's see. Well, this is a schematization of the surface water in Berlin. It's a project I've been doing together with a lot of people in Europe, where Berlin was one case. And we also looked at the Danube, so one of the large rivers in Europe, and in groundwater and a number of other things. Actually, quite a large variety of systems. And what I did for Berlin was, let me get the picture. Yes. So what you saw here, this fairly complicated surface water system, I turned it into a Deltares schematization like this. It's it may be a bit difficult to recognize, but I try to capture the main features. And there are a number of lakes inside Berlin. This is the Müggelsee. They have the Wannsee over here, the Tegersee. It's a bit small, of course. Oh yeah. But using the data I had on the hydrology of that, I was able to set up a model for simulating the transport of PFAS in this whole water system. Problem is that we didn't have much data on the PFAS in the water system of Berlin. So we had to make some estimates. But well, this is an example of using Delwick without an actual full-scale hydrodynamic model, as is the other one. And as it happens, this project called Promises is going to be finished next week, and I have the pleasure of going there in the middle of France, in fact, in Orléans.
Arjen, there's also a question. Is the water quality module only available for certain contaminants? How accessible is it to customize for a unique chemical?
What Delwick or the water quality does is it will consider any substance. You can give a substance a certain name. It will consider it a conservative substance unless you specify processes for it. For instance, nitrate can be absorbed by algae, but if you don't include algae or that process, then it will be a conservative substance. It doesn't really matter what kind of substances you do. The specific characteristics of a substance become defined by the processes that work on them. If you look at say organic micropollutants like PFAS, they can absorb to organic material, and that organic material can sink to the bottom. So that would be a flux of PFAS towards the bottom. That can be modeled by a process where you have partitioning between PFAS in the water and PFAS on organic material. And then if the organic material sinks to the bottom, then you make sure via a process that that absorbed PFAS is also going to the bottom. So that's something that has been included in the model. For organic micropollutants, we have a small number like PCB 153 or hexane, that sort of substances where we have fairly detailed data about adsorption coefficients and volatility and that sort of things. So there you have specific processes. On the other hand, there are so many organic micropollutants that, as long as you can mimic the micropollutant of your interest, using any of these more or less generic ones, it's a matter of supplying the right process parameters that describe the behavior of that substance, and then you can do the calculations. So it really depends on what kind of substances, what kind of characteristics you want to include. Whether it's easy because it's already there, or because you only have to make some small um, adjustments, or whether it's going to be much more work because what you want to do is quite new. I can show you the substance files. I didn't do that so far, but the substance files are in fact simple text files that have a few conventions, and these names, in this case oxy for instance, that's a name that occurs in the process configuration, and Deltares then can tell, okay, oxy, that's a process, that's a substance that is subject to a number of processes, and these processes are selected over here. So there you have, let me show that. There you have the names of the processes that you want to include. Of course, it's a bit much to demand from users that they know all these processes, and for that we have a small tool called the Process Library Configuration Tool, which allows you to set up these substance files. But you can also edit them yourself, and if you need to configure these processes, well, there is another way to do that. Let me see whether I can find that quickly. What we use is typically this kind of setup. This is a configuration of a process which calculates the pH based on carbonates. Here you define the inputs. Here you define what comes out of that. And then there are a few things like the fluxes that we, as we call them, that define how much of one substance is turned into another substance. It's a bit much detail to explain right now, but this is the way we have set up our process library, and we have tools to maintain this. So these are also part of the installation. So you can extend the program as you see fit, if indeed it's necessary. Are there any specific substances you would be looking for?
Yeah, I don't know if Brian's gonna respond to that, but I can say that as a user, like there's a wide range of substances available, and usually what you're interested in modeling is like similar to something that exists already, and so you could copy that and you're just changing kind of the decay coefficients or the partition coefficients, or the different kind of parameters that are setting that. And then it's a relatively straightforward transition, even if it's not something that's explicitly not explicitly included as a substance. Likely the way that you would represent the substance is in there.
What I want to show here is HCB, so hexachlorobenzene. It is defined via a number of coefficients. Most importantly, the partition coefficients over here. So we have a number of such substances. There's also the more generic one, organic micropollutants. Let's see whether I can find that. Maybe not the most happy organic micropollutant then. Cool. Well, you can see there's a here. There's a number of such organic micropollutants in the database. They are involved with a number of processes, so you will see that these names occur a number of times, but it is quite possible to specify new values. What I should add here is that if you are not satisfied with a value like this is the default value. You can specify the value for this particular process coefficient in the input, and then it will use that instead of this default. That's the way the process library works, and you get a nice. Let me see. You get a nice listing of all the processes that are involved with their with the substances and so on, and what kind of values are used, either default values or the values that you specify yourself. So in that respect, it's quite quite flexible. And what we also sometimes do is if we have a substance which behaves more or less like this, but it's not quite the same name, we abuse a name. It's not the best way to do it, but it certainly is one way to handle new substances. And like I said, if necessary, you can also add. Oops, I see my laptop is running out of. Okay, well, 29% still. But yeah, if a substance is completely new, then it's certainly possible to add new processes. Something we did not too long ago is we added terrestrial vegetation to the whole system, and that's something that's completely different from a transportable soluble substance like nitrate or whatever.
We're getting some more questions. Someone asked, what species of bacteria indicator, I think it might be indicator bacteria, can you simulate in your model?
Okay, what we have predefined is well, Escherichia coli, so coliforms, both total, fecal, and E. coli. It's always a bit difficult what kind of species you really are dealing with. If you have a species of bacteria which behaves or is subject to the same sort of processes as, say, coliform bacteria, you can use that to model, well, this other kind of bacteria. I think we do have streptococci separately. No. Well, you could use E. coli with different coefficients to model streptococci. In fact, I included viruses some time ago, just using the same process we already had. And the viruses, well, they will decay slowly, but they will also absorb to organic material. But based on the information we had on the kind of the kind of processes they are subject to, it was possible to reuse all the various process routines and simply configure that to include viruses.
There's also a question if you have an example, like a calibrated example, where you can show that the model performed well against collected data.
Right. That would be part of particular projects. I can dig up a few of these. I mainly try to solve the problems with the software itself, so I don't have many shiny examples, at least not ready. But I can certainly try and find them. I've made an action point of that.
There's also a question about, can you use any other mechanism other than the constant decay for bacteria or virus? So I guess they're interested in what the bacteria process looks like.
Yes, you certainly can.
So maybe.
As a matter of fact, what we have included is the influence of salinity and solar irradiation as a function to influence the rate at which these bacteria decay. I know there have been studies into the possible aftergrowth of bacteria, of these coliform bacteria. I don't have any information about that. Those were studies from long ago, and if that really occurs, then that's something that would have to be included in new process routine. But well, if you have information about that, we can certainly do that. I can try and see what we have on bacteria in the documentation. Right.
Yeah, I mean, I was gonna say, yeah, I remember it having some sort of sorption or soling.
Yeah.
Or something.
Well, what you can
I
see
don't
here
remember the details.
is the extinction coefficient. That's important for the light.
Okay, yeah, they have absorption and sedimentation. That's what I remember. Yeah. Okay.
So that sort of aspects are included in the library. I have a question for you. We've been talking about Lake Champlain, and Leaf, you showed me the layout of that lake. Could you indicate where the typical water quality problems arise and what kind of problems they are?
Well, I could kind of. I mean, so one of the things, they're all over. Here, I could share. Can I share this PowerPoint? Oh, this is
We
a
stopped sharing.
presentation that I'm working on, so it's not complete yet, but I can show you that. And I would look. I haven't. Whoops. But yes, there's lots of. Where was I? Oh, so this is a this is a map showing all the places that blooms were
Yeah.
bacterial blooms were observed in Lake Champlain. So the water quality problems are actually all over. And I was looking for my presentation that had some at least calibration of the temperature stuff, which is actually done in the hydrodynamic model, but just to show like sort of, how well the model performs, at least in terms of temperature and current, that the hydrodynamic model can perform quite well. I have some plots from the model there, but I am not finding them quickly. But yeah, there's a lot of so it's all over. So we have a hydrodynamic model that performs reasonably well for the lake, and so yeah, we're interested in extending, adding some more direct water quality modeling to it, possibly. And there, you showed the nitrate diagram, but of course in Lake Champlain, the issue is phosphorus, but it's a similar set of equations.
And what kind of cyanobacteria do you observe there? Is that Phaeocystis or
I don't
is
know.
it
Anthony, do you know about the species? I don't know the details. But I don't know about the culture. I'm a physicist.
okay? Well,
So are you, which is why we get along so well. But
yeah, well, we have had problems with floating algae, also cyanobacteria in some of the lakes here in the Netherlands, and we've tried to model these layers using fuzzy modeling. It wasn't really successful, but the data we had was also very fragmentary and almost anecdotal. So once the managers of the lake would say, "Well, yesterday we saw this layer, this green stuff on the lake," and yeah, well, that makes it very difficult to quantize the effects of quantify the effects of these cyanobacteria, etc. Is that something that happens in Lake Champlain also, or is it too large for that sort of thing?
yeah, I don't know. I'm sorry. I can. I did find. Sorry, I was going to show briefly. Yeah, I have the model on Lake Champlain. And I was just gonna. I can skip to showing that the underlying. Oops, we're just gonna skip through. Is there a way to? There's a lot of detail that goes into the hydrodynamic modeling, but we're able to measure the water levels really well. And then also in terms of water quality, we get the temperature across the whole depth reasonably well. This is showing a simulation in the main lake, so it's at a quite deep part at like 43 meters of depth. And we, the model is able to accurately capture the depth, average temperature, the temperature is close to the surfaces we have, and the temperature depth, and then also capture the stratification, which are all like
right away.
important physical processes, are important to capturing the
Because
downstream
the battery, I'm going to move to another office.
Okay.
Yeah,
I mean, I think we're running out of questions here. I don't know if Anthony, is there anything else? Are there any more questions?
If you have questions later on, then please let me know. I'll be away for a few weeks, so back in the second week of May. If I can't answer your questions tomorrow, but don't hesitate to send them. I'll be happy to send more information or explain more.
Okay. Thank you very much.
You're welcome. It was quite pleasant to do this. Yeah, of course. I'll type my email address. There you go.
All right. Thank you so much, and thanks everyone for coming. And hopefully we'll see you at some more of the water quality group modeling seminars.